This is the protocol for the sample preparation for the plasma assay. Please note, to ensure the integrity, all samples and standards should be kept at 2 to 8 degrees Celsius during collection and processing. Once handling is completed, store all samples at -20 degrees. For the standard preparation, prepare 2 independent stock solutions of each analyte, ivacaftor, M1 metabolite, M6 metabolite, and lumacaftor in methanol at 100 microgram per milliliter and 10 microgram per milliliter. Please note compounds expire after 10 days on bench top. For the standards, transfer 100 microliters of human plasma into the 1.5 milliliter polypropylene microcentrifuge tubes. Continue to do so for all your other samples. Then add the internal standard to each tube. For example, we choose 0.01 microgram per milliliters out of the prepared standard. Now this tube contains plasma spiked with the internal standard. Proceed like this with all other standards. Take your standards and spin it down for a few seconds to make sure that all liquid is at the bottom of the tube. Continue to do this for all your 40 standards. For the patient samples, transfer 100 microliters out of the patient sample tube into the microcentrifuge tube. The real patient sample is not spiked with internal standard.